Hey there, listeners. Welcome back to the Let's Get Oral podcast. I'm Jonathan, one half of the LGO crew. In this episode, we continue our IDM Totality of Care series by sharing a conversation Camille and I had with Dr. Nathan Bryan, the keynote speaker for this year's symposium. Nathan is an international leader in nitric oxide biochemistry with a collection of patents, publications, and personal care products to show for it. So, without further ado, here's our conversation with Dr. Bryan at the 2024 Totality of Care Symposium.
Let's get oral. Let's get oral.
All right, we're here again at the Totality of Care in Knoxville. I have with us today Dr. Nathan Bryan. I am so excited. I have been following your work for a very long time, mostly in the chlorhexidine space, but I would like to start out with your history in nitric oxide research. What brought you to the oral cavity?
We have to go back probably 20 or 25 years. I was a student at LSU School of Medicine working on a PhD in molecular and cellular physiology, and the Nobel Prize was just awarded for the discovery of nitric oxide. And Lou Ignarro, who had just won the Nobel Prize, came and spoke before the student body. And, he told his story of discovery and the Nobel Prize and this molecule, nitric oxide, that I'd really never heard of up to that point. So it was fascinating. It's a gas, it's a signaling molecule, and, there were a lot we didn't understand at the time. And really, my PhD work was developing sensitive and selective analytical methods where we could detect this molecule in biological samples. So nitric oxide, when it's produced, it's gone in less than a second. So how do we quantify and detect this in different patient populations? So that really allowed us then we developed some sensitive methods, gas phase chemiluminescence techniques where we could detect this gas in the exhaled breath and in tissue biopsies and things like that. And then that really provided us the tools that no one else had. And we were publishing 10, 12, 15 papers a year during the heyday. And then we started to uncover kind of the role of the microbiome. There was a lot of work done in the early 2000s on mapping out the entire microbiome that started in the gut microbiome. And then, of course, it spread to, the skin, the oral cavity, and really every orifice where there's a microbiome. And then there was data. We looked at the literature and found that in the early 90s, there was data showing out of the UK and out of some guys at the Karolinska Institute that there were some oral bacteria that were called nitrate reducers that would metabolize the nitrate found in vegetables into nitrite and nitric oxide. And if you eliminated this, you seemed to disrupt what we were looking at as markers of systemic nitric oxide production. So then in collaboration with the dental school at the Texas Medical Center, where I was a professor of molecular medicine, we started doing some studies and just trying to, number one, can we culture these bacteria? Are they culturable? And then could we identify certain patient populations that maybe were lacking these bacteria and that have any correlation to their nitric oxide output and their blood pressure? And, it was fascinating because we could culture these bacteria, but in some people we didn't find any of these bacteria, and they seemed to have the highest blood pressures.
Yeah.
Then we moved it forward and actually did more controlled clinical trials where we only took people with normal blood pressure and then gave them mouthwash twice a day for 7 days, did daily tongue scrapings, monitored their blood pressure, and found that if all we did was eradicate the oral bacteria, we could affect their systemic blood pressure. And that was a completely new paradigm in regulation of systemic blood pressure. How are the oral microbiomes affecting, vasomotor action and blood vessel vasodilation and regulation of blood pressure?
Did you get some resistance to your work at that time?
It was exciting because we really pulled, the periodontics division of the UT Dental School. We pulled in the entire metagenomics division of Baylor College of Medicine that was doing the analysis and the full metagenomic analysis of the bugs. And then, in the Department of Molecular Medicine, we were trying to correlate that to human disease.
Yeah.
So it was exciting, but when you start to kind of interfere with what daily practices are in oral hygiene and really the dental community, then you got a lot of pushback because this is completely new. Because dentists, if you have a pathogen, an oral pathogen, they want to just throw an atomic bomb in there and kill everything, right? So now we're trying to figure out how do we selectively kill the pathogens while maintaining the good guys.
Right, exactly. At what point did you have your aha around all of our oral care is really predisposed on antimicrobial approach?
I was, when we first published those papers, maybe 15, 20 years ago, I was invited, and really my first introduction into the dental community was, I think, an organization called the IOAMT, the International Organization of Mercury Toxicity, or I can't remember what the acronym is. And so these were
that feels very adjacent.
Yeah, no, but it's these were kind of the dentists that were on the forefront of kind of thinking differently.
Right. Okay.
So they were against mercury amalgam fillings.
Yes.
They had a little bit different thought on root canals.
Yeah.
And how that was affecting systemic health. So once they got word of what we were doing, then I became invited to speak and present our data at the dental events. And then I was invited by more dental events because they're part of different organizations. And so that group really adopted what we were doing because it was within kind of their realm of possibilities and kind of the way they were looking at oral health. And now we're moving in and hopefully trying to make it more mainstream because it's not just our work, it's the work of many groups now trying to figure out that it's not just the presence of infectious pathogenic bacteria, it's a disruption of the non-pathogenic commensals. So I think, I heard a line in there that I really resonated, that we have to change the focus from treating disease to kind of restoring health.
Right, exactly.
And the microbiome is an important part of the human host health. So we destroy it to our own demise, I think.
Well, it's an interesting ecological niche, and you can think of any ecological system where if you get rid of one thing, there's going to be something else that has to come take its place. But also, I've been working in the inflammation cascade. The inflammation drives certain metabolic changes in that community and causes dysbiosis. So there's a lot happening kind of reflexively throughout the day with circadian rhythms and inflammation cascade that is normal and part of mastication, for example. I mean, there's all this impact in that oral cavity. But I wanted to kind of get back to your research where you did a knockout mouse model to show that it was indeed mostly the oral microbiome at play. Can you talk us through that study?
Well, in the basic sciences, we have the benefit of doing, gene knockouts or gene insertions and looking at the structure and function of certain proteins. And so in the enos deficient mice, so you can actually remove the enzyme nitric oxide synthase in the lining of the blood vessels, in the endothelium. In these mice, phenomenally, they develop, their first phenotype is insulin-resistant type 2 diabetes. They develop an increase in blood pressure. They have hypertension. They're more prone to injury from heart attack, stroke. So it really recapitulated what we thought about production. But we can completely rescue these mice simply by giving them nitrate in their diet and maintaining, a healthy microbiome. You completely rescue the hypertensive phenotype, the diabetic phenotype, simply by capturing the activity of the oral microbiome and feeding them a diet. Fortunately, in mouse studies, we can control their diet, we can control their light cycle, we can control their microbiome. And that's the reason that most things never translate into human medicine because, number one, everybody has a different microbiome, every person has a different diet and have different oral hygienic practices. So now you take all these confounders that we controlled for in the mouse studies, and now you just, it completely disrupts the experimental design. But I think now what we're finding is, and we've done this, we published this, I think, in 2014, if we give those mice now the chlorhexidine and kill their microbiome, then you completely eliminate the protective benefits of the diet, and you basically overcome we no longer can protect them from their enzymatic deficiency through diet and the microbiome. Right. So this tells us that there's a balance. You can, if you have a good endothelial function, then the oral microbiome, I think, still plays a role. But if you develop endothelial dysfunction, you can't make nitric oxide. Now we're 100% dependent upon these bacteria.
So what kind of pathologies align with endothelial dysfunction?
Well, endothelial dysfunction is now recognized as probably one of the earliest events in the onset and progression of all chronic disease.
Right.
Whether it's First one is usually sexual dysfunction. When you lose the ability to regulate blood flow to the sex organs, you can no longer get dilation, you can't get engorgement. And then it's hypertension, it's metabolic disease, it's autoimmune disease, because nitric oxide is what kind of releases the brakes or kind of puts the brakes on the inflammatory cascade and inhibits things like NF-kappa B, these interleukins. So if you lose the ability to produce nitric oxide, now you get runaway inflammation, which obviously leads to a number of different disease cascades.
So now that we have a really nice provocative study in mice showing that we could potentially solve our problems through diet, what does that look like in practice?
Well, on paper, it's quite simple. Practically, it's very difficult and complicated because, our whole question was once we kind of made that observation, we go, okay, how can we modify current dietary guidelines and give humans enough of these nutrients to where they can regulate their own blood pressure? And so we went to 5 cities across the U.S. and we just measured different vegetables, I think 5 different vegetable categories in New York, Raleigh, Chicago, Dallas, and Los Angeles. We brought it back to the lab and we found that there's as much as a 50 to 80-fold difference in the nitrate content of celery grown in Dallas or Los Angeles opposed to, Chicago or New York. So there's regional differences in agronomy.
Okay.
So in the South, in this Rust Belt, where there's a lot of lightning storms, the air we breathe is 78% nitrogen. And to break that triple bond of N2, it takes high energy, and it's usually through lightning. So in areas where there's a lot of lightning storms, the soil is typically more enriched in nitrogen. So now the vegetables grown in that soil assimilate more nitrate, and then we can accumulate more of that as vegetables. And then, the other interesting thing was we compared conventionally grown vegetables to organically grown vegetables.
Oh, and?
Found that, organically grown vegetables have about 10 times less nutrients than conventionally grown.
Why do you think that is?
Well, to get an organic label in the U.S., there's certain things you have to, so there's no herbicides, no pesticides, but you can't add nitrogen to the soil.
Okay.
So without a standardized form of nitrogen in the soil, how can you standardize the nitrogen? I mean, you can add manure, compost, things like that, but again, it's highly variable.
Do you think that the data from the dietary studies is wildly flexible, and it could be because of the source, but also how does mastication play in?
Look, I think it's very important because there's the bacteria in the mouth, and what we're interested in are the dorsal part of the tongue and the rear part of the tongue and these facultative anaerobes. So there's a certain resident time that the food has to be present in there. So I think it's important to chew your food for a long time, really allow that. But what we're finding is that the mastication is the first part. But then when we chew our food, we swallow it, it goes in the stomach, half an hour or so, give or take, get gastric emptying. But then there's transporters in the duodenum, in the proximal gut, that then take the nitrate that's from our food and then concentrate it in our salivary glands. And this takes about 90 minutes.
Okay.
And now for the next 6, 8, 10 hours, each time we salivate, we're getting secretion of nitrate into the saliva. Now these bacteria can utilize it and respire. Then they perform that 2-electron reduction of nitrate to nitrite. When we swallow our own saliva, it becomes protonated, and we make nitric oxide in the lumen of the stomach. So that's the cycle that has to be present. So if we're not getting enough nitrate, it doesn't work. If we don't have the right oral bacteria, it doesn't work. And if we're using antacids, it doesn't work.
Could you speak further about who are the key nitrate and nitrite reducers in the oral microbiome that we should be keeping
there's
an eye
about
on?
maybe half a dozen to a dozen major kind of genus and species of bacteria that we've identified that are cultured that seem to be really necessary and sufficient for this nitric oxide production. But it's Haemophilus, it's Vianella, it's I'm drawing a blank, but we published on this, I think in 2012 and 2014, and then again another paper in 2019. But there was really 4 to 6 ones that we could predict steady state blood pressure in all these patients if they had these bacterium. Not only were they present, but the numbers in which they were present.
In periodontal disease, are these bacteria present or reduced or increased? Do you know?
That I don't know. Well, at least kind of on the surface, we haven't delved into this too deeply, but people with oral dysbiosis, whether it's periodontal disease or gingivitis, there's a disruption in the diversity.
Yes.
So, and what we're finding is that in those people that we really, and from the first study we published in 2012, I believe, that there was no culturable bacteria in the people who had the least diverse oral microbiome. And they had a lot of these pathogens present. So at least there was a correlation. So there was an association. But now we're trying to figure out, okay, if we can restore the normal ecology, do the good guys keep the bad guys at bay? But what's clear to me that if you destroy the ecology of the microbiome, it allows for these opportunistic pathogens to pop up, cause disease.
Right. Well, I mean, and I'll talk about this all day, but Porphyromonas gingivalis, most widely studied oral pathogen, we have demonized it for a lot of good reasons. You know, when it's in abundance, it's no good. But when you have the inflammation, which produces all of these cytokines, peptides that are all inflammatory, those now become food for Porphyromonas gingivalis. And so what's happening is when it gets a whole lot of that type of food, it's like the rest of us getting the wrong type of food. We turn into like, I'm going to have a poor diet. I'm going to be grumpy, right? And I'm going to do bad things. Same thing, same principle. And so you shift its LPS phenotype, you shift all the metabolic activities, and it becomes this more pathogenic feature. And I still maintain that, you know, we see it in healthy people to a very small degree. And I suspect that degradation of inflammatory, that property itself is actually possibly in our favor when it's in the right conditions. And that the type of LPS that is without the inflammation is possibly to our benefit immunologically. And so this is where I hesitate calling anybody a pathogen, because it's only a pathogen when everything is going wrong, right? Right. But certainly with the nitric oxide F-nuc, for example, we have always kind of put in even though it's part of the purple group of the Sokransky complexes, we've kind of coupled it with the red complex because we know it's an interim bacteria that allows those pathogens, periopathogens, to stick. But now we know that it also helps the good bacteria that we need for this nitric oxide breakdown. Mm-hmm. So again, we're finding that we have these periopathogens for good reason, and that I would like to shift the discussion or, demonization, if you will. Because again, I think that's why we have antimicrobial oral hygiene as our primary care, and now we're realizing, it's part of our metabolic health. Well, poop. Yeah. Oh no. But so now we have to kind of get back to how do we balance this? Like you said, going in and picking out the pathogens or creating a balance to the mouth that we didn't have. What's out of balance? Yeah,
I think let the bugs themselves work it out, right? 'Cause I think that as long as you have the good guys, they kind of keep the bad guys at bay. But
and get
when you
that
destroy that
inflammation.
biofilm and the ecology, then it's a free-for-all.
Yeah. Absolutely. One of the most fascinating findings from my dissertation work was, let me take a step back. I developed a in vitro biofilm model, but a complex one that supported your strict anaerobes, Candida, your keystone, streptococci, et cetera, et cetera. And what was fascinating to me was if you removed the Veillonella, it just collapsed and allowed your Candida, your Porphyromonas, and some of these other red complex bugs to take over that model over a matter of days. So I can only imagine what that looks like if someone is using antimicrobials 2-3 times a day in their mouth. I love Camille's analogy of, if we're treating our mouth like a toilet, right? Right? If you keep bombing it, how disruptive that is, for the diversity of it all, and then how that cascade can lead to not producing enough NO due to losing the members who are able to convert that nitrate and nitrite. Sure. And then further downstream too, what's being done when it is reintroduced into our salivary glands. Because that's a part of the story that even I was lost upon until more recently, and it was oh man,
yeah, right. How do we replace it? So once it's gone dysbiotic, and I live in New York, so my produce isn't as amazing.
That's right.
So what am I doing now? Do I have an oral care hygiene product now that I get to deliver through my gum tissues, or do I still have to find a way to dietary replace? What am I doing?
Well, I think, look, it's not just in New York, there's different. I mean, if you look broadly, since the 1940s, today the food that's grown in America has 78% less nutrients than it did in the 1940s, and that's just the farming practices, the lack of crop rotation,
Right.
the overuse of pesticides, herbicides, depleting the soil. So I think everybody has to supplement to some degree.
Yeah.
And that's why I recommend, do micronutrient analysis, figure out exactly what your body's missing, and then give it back in form of supplementation.
How do you evaluate that for yourself?
There's blood tests where you can do micronutrient analysis that'll give you most of the basic micro and macronutrients that the human body needs, and then whatever you're deficient in. It's really simple. So if you take your vitamin D levels, blood levels, and they're 50, you titrate up and you supplement up till you can get them up to 80.
Right.
So those are healthy levels, but I think we have to know, right? We can't make assumptions. But I think there's really no way to know if you're getting enough nitrate from the food you're eating, and that's what we've been trying to solve that riddle for. How do we normalize this? How do we give the body back what's missing? How do we restore normal nitric oxide production, not just through the oral microbiome, but in the lining of the blood vessels, in the neurons? How do we activate our immune cells and lead to adequate nitric oxide production when we get upregulation of INOS to kill the infections, to
Right.
mitigate inflammatory response, at least the acute or
Right, and hopefully it's not chronic.
Yeah.
So what do you think of the salivary strips for evaluating the nitrite in the saliva?
Well, I first developed those in 2009. I abandoned the patents on them, I guess, in 2012 because it's old chemistry. It's called the grease reaction. We've been using that for hundreds of years. But we thought we could have a method of use on it. But, I think they're a good engagement tool.
Yeah.
There's no false negatives. So if you're negative and if you don't light up that test strip, then there's no false negatives. What we worry about are false positives.
Yeah.
And that's why I don't use them anymore, because people, if you have an active oral infection, then
yes.
what we're testing in the saliva is a local immune response
Exactly.
in the gingival
That's
tissue.
right. Mm-hmm.
And so it's certainly a false positive. And so they may be an active immune system and nitric oxide being produced from that infection or inflammation, but systemically they're completely depleted. Because what we found, I think we published this in 2004, that people who have chronic inflammation, there's an upregulation of INOS,
Yeah.
and it leads to a feedback inhibition on the constitutive isoforms.
Got it.
So if INOS is upregulated for a long time, it shuts down your constitutive isoforms. So we've got to mitigate that inflammatory response, maintain normal constitutive NO production.
So you really do need a full panel of information. You need the CRP you need all of to get the full. I gotcha. That's fascinating, and especially since 90% of the world has some sort of oral inflammation, that's a pickle.
No, that's right. And that's why I don't, I think, again, there's a number of companies now that use it, that sell it, and people ask me all the time. I mean, you can use it, but you have to understand the caveats and the nuances of what we're actually detecting and what that means. Yeah. So you can interrogate that pathway if you know what you're doing and how to use those strips, but simply naively going in and, we saw it all the time. A 50-year-old obese, diabetic, hypertensive man with erectile dysfunction comes in and he tests positive on the test strip. He goes, "Oh, I don't need this."
Right.
You go, No, you do. So then, obviously, we can do a full oral exam, and he's eat up with, pathogens and got oral dysbiosis, and so that's his problem.
So Dr. Glogauer out of University of Toronto is hopefully finalizing his FDA-approved device, which would be a chairside salivary diagnostic 10-minute assay to show inflammation. So this is basically an artifact of neutrophil biology to show a gradation of intensity based on the neutrophil activity. So hopefully, I'm hoping that will come forward and be more readily available. He promises to keep it affordable. So.
Yeah. No, I think we're moving in that direction. Obviously, we've got a history of developing products, nitric oxide-based products, consumer products. We've got drugs now going into phase 3 clinical trials for drug therapy for things like ischemic heart disease, Alzheimer's, and a topical drug for diabetic ulcers. But for the past 2 years, after it goes back further than that, once we started recognizing the dysbiosis in patients and starting to ask this question, because, of course, the first question was, well, how do I fix this?
Yeah.
Because my dentist, I've got halitosis or I've got severe perio or gingivitis, and my dentist wants me to use chlorhexidine or mouthwash every day to keep the infections at bay. So then the question was, well, how do I do this? And the answer was, you really can't.
Yeah.
I mean, so you have to, it's risk versus benefit. So then that started to think the conversation is there a way we could, through an oral product, whether a toothpaste or a mouth rinse, where we could feed these good commensals, restore their population, improve the diversity, while keeping some type of antiseptic effect on the pathogens. And I think we've solved that. I mean, we had some early prototypes the last couple of months. We've sent it out. We've done market focus groups and market research, and it appears that we've cracked the code on that. So
That's
we're
incredible.
going to be developing a toothpaste. Will come to market probably in the next 90 or 120 days. Then we have a mouth rinse that's not antiseptic, but the early data indicate that we can restore and enhance the diversity of the microbiome. And best case scenario, and we're still doing the testing, but selectively kill off the pathogens. The things like the PGs and the ones we worry about.
Can you tell us about the technology, or it's still
Well,
too early?
It's still too early.
Okay.
We have some early prototypes that seem to be that we probably don't have to do that much tweaking anymore. But everybody we sent it out to in our focus groups loved it. Loved the feel of the toothpaste, how it left their mouth. We think the next clinical study we'll do is just using the toothpaste in the mouth rinse. Can we take resistant hypertensive patients and normalize their blood pressure?
Yeah.
Because we've clearly demonstrated that if you destroy the microbiome, your blood pressure goes up. Now, in patients who have resistant hypertension that are resistant to drug therapy, can we restore the oral microbiome, restore nitric oxide production, and normalize their blood pressure? If we can demonstrate that, then this completely changes dental medicine, and I think really cardiology as we know it.
Yeah. Incredible. Well, I'm going to leave it here so that we can get you back when you release it.
Yeah.
'Cause that's
We still
exciting.
got a lot of work to do.
Yeah,
But I'm
I'm
excited.
sure. Thank you so much for sitting down with us today. This is brilliant.
Thank you. Thanks for
So grateful.
enjoyed the conversation. Thank you guys for what you do.
Thank you.